You found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on this.
Hey there, everybody. Dapper Dinosaur here. I am here with Dr. Dan Stern Cardinal. So, Dr. Dan, why don't you give yourself a quick introduction for the few of my audience who might not know you?
Hi, I'm Dr. Dan Stern Cardinal. The internet apparently now knows me as Dr. Dan. I run the channel called Creation Myths. I'm an evolutionary biologist, and my specialty is viruses and microbiology and genetics. So yeah, I think that's good for now. If for some reason you're watching Dapper Dino's channel and you're not already subscribed to me, come on over. We do a lot of the same kind of pseudoscience debunking stuff. So come on over and say hi.
Absolutely. Check the description for a link. And if I remember, I'll also put a link up at the top right. So, of course, will I remember? Who knows? Now, this paper is in reference to a claim in Islam from Sahih al-Bukhari 3320, or using the other reference method, Book 59, Hadith 126. It is narrated Abu Huraira. The Prophet said, "If a housefly falls in the drink of any one of you, he should dip it in the drink and take it out, for one of its wings has a disease, and the other has the cure for the disease." So, the claim here that we're going to be seeing a defense for is, let's be clear, houseflies carry a disease. So each one, I suppose, could have a different one, but at least one disease is going to be on one wing, then the other wing is going to have a cure for that disease. So this is the claim we are testing here. And so we are reading microbiological studies on fly wings, Musca domestica, where disease and treat, and that is as written. I want to say first, there is going to be some iffy English, and I don't want to make that too big of a deal because I'm very sure that the author, Raheb Mohammed Atta, is not a native English speaker, and I would say, however, that the World Journal of Medical Sciences, which is where this is published, should probably get a better editor, maybe one who's a native English speaker.
Yeah, why don't you get us started? I have because a lot of this, I will say, a lot of this, the abstract and the intro, a lot of it has, like, it references religious stuff that I'm not familiar with.
Right.
And then it kind of, once we get into the methods, that's where I can really comment. When we talk about the collection of the flies, the microbiological examinations, that's a lot of these techniques are actually things that I have personally done.
Okay.
So I can look at these pictures, for example, and say, like, does this check out? So that's where I'm going to be most useful here.
Over 1,000 years ago, Prophet Muhammad, peace be upon him, said, "If a fly falls down your vessel, drown it, then remove it, for one of the wings has the ailment and the other has the cure." Narrated by al-Bukhari, even though it's actually really Abu Huraira, and Bukhari collected it, whatever. That's not important. In this study of 4 flies of Musca domestica species were collected to investigate the antibacterial and antifungal activity of their wings. The obtained results show that all media cultivated with right wing extract were free of bacterial and fungal growth. However, bacterial and fungal growth for the left wing were observed. It could be concluded that the fly right wing is considered as a new revolution of antibiotic, recommended for further researches to find more antibiotics from right fly wing. It's, like I said, English is a little tough to get through, but I think we get the idea here. So, allegedly, we're going to find evidence that the right wing of flies have antifungal and antibacterial properties that the left wings lack, which would be pretty significant. And also, I would say that would be hard to explain in sort of an evolutionary sense. Like, why would a fly have a left to right distinction in its, disease inhibition? Because presumably, if the fly has these properties, it's doing it to keep itself from infection. So why would it be like, oh, only my right side needs to be protected from infection, left side, whatever, who cares?
Right. It does. It is challenging to find a biological basis for how that would operate. Yeah.
Right. Oh, by the way, the key words for this are fly hadith and new antibiotic and housefly miracle. We are already apparently taking the stance from the abstract that this is apparently miraculous, which I guess could explain why there would be a far-fetched biological process going on here. If it were miraculous, I guess I could say, well, sure, I guess I would explain why evolution would have a hard time figuring this one out. So, the introduction we have, there are so many scientific facts that have been proved in both the 20th and 21st century that confirm the hadiths of the Prophet Muhammad, peace be upon him. So I want to quickly explain what a hadith is. So most people know what the Quran is. The Hadiths are another source of theology and jurisprudence in Islam, and they are the collected alleged sayings and deeds of Muhammad, and so they form our most important biographical source on Muhammad. The way these work is that various people passed these down orally until they were collected a few hundred years later, and Muslims generally judge the reliability of these hadiths based on the reliability of the chain of narration. So this guy said to this guy, who said to this guy, who said to that guy, who said to this guy, and each person in that chain has some like determined level of reliability. And if they're all reliable all the way down, then it's considered to be a reliable hadith, and it's usually judged by the weakest link in that chain. So whoever is the least reliable, that's how reliable the hadith is overall. Now, I do have, as someone with a history degree, some problems with this technique for judging reliability of oral tradition. I don't think it works very well, but that is not why I'm here, so we'll just leave it at that. I'm okay with accepting that this is the claim that Muhammad made. I don't think it matters much. I just want to explain the background there. It continues, where 1400 years ago, where there were no science, labs, or technology to solve many scientific facts, to focus on today is a hadith that the Prophet Muhammad, peace be upon him, told us about the fly and how there is a healing exactly. The hadith narrated by Abu Hurairah, Allah's apostle, said, "If a fly falls in the vessel of any of you, let him dip all of it into the vessel and then throw it away, for in one of its wings there is a disease, and in the other is a healing antidote for it," i.e. the treatment for that disease. Al-Bukhari and lists reference 1, but we already gave the reference, so. Hundreds of thousands of flies types exist in the universe. However, just 10 types share us living in our houses. The female fly lays down more than 100 eggs in the dung of some animals or in the garbage. After one day, the larvae come out and it feeds from the surrounding environment. 2 weeks later, they become full-grown flies. In the 4 generations, one fly can lay down 1.5 million eggs, but fortunately, the majority dies due to weather circumstances or becomes food for birds and other insects. Maximally, a fly can live for 60 days. Okay. The last thing that might come to mind is that we can find antibiotics on the surface of flies. Okay, so I want to comment on that. That's actually not the last thing that would come to mind for me, because flies live in very highly contaminated environments, and I would actually expect that flies would have a very robust system for avoiding bacterial infection. What do you think, Dan?
Yeah, that makes perfect sense. And it's not just for things that live literally in dung. It's like everything, like on our skin. We have antimicrobial peptides. We have acidity. We have, and all the openings in our body. We have mucus. We have like all these mechanisms.
Yeah.
They're called barrier defenses that keep bacteria that are outside of us from getting inside of us. It's a common feature of animal life, plant life. Basically, if you're multicellular, you have to have a way of keeping bacteria that could potentially parasitize you outside of you, right? Just this kind of outer level of defenses. And some of those are going to be physical, like just skin or an exoskeleton or something. And some of those are going to be chemical, and that can be something like chemicals that we would call like antimicrobials or antibiotics. Or it could be something simpler than that, like just mucus that stuff gets stuck in, or acidity that's makes an environment that's inhospitable for the bacteria. So this isn't just a fly thing. This is an every animal thing. There has to be a way to keep bacteria on the outside.
I mean, I would go farther. It's basically every macroscopic organism.
Yeah.
The bioscientist Joanne Clark observed the fly's resilience, decided to study flies as a possible source of drugs to combat deadly bacteria. Ms. Clark research showing that flies produce their own antibiotics will lead to better treatments for human infections from Escherichia coli, E. coli, and other virulent bacteria, even perhaps Staphylococcus aureus, S. aureus. Infectious disease kills at least 17 million people annually. Previous results is being supported by global pharmaceutical company GlaxoSmithKline over the next 6 months, trying to isolate antibiotic compounds from the material collected from the flies. These days, Russian doctors observed that flies contain many substances that can be more effective using bee handling than traditional medications trying to develop treatments using flies. This new treatment will lead to a revolution in medicine's world. Entomologists' latest research certify that there is very little difference between a human and a fly heart, where... Okay, hold on. Okay, so that's the thing that I want to talk about. So here's the thing. A fly's heart is basically just a tube of muscle that contracts.
Yeah.
And it doesn't have chambers. It doesn't lead to blood vessels. Flies have an
It
open
does.
circulatory system.
It does lead to vessels, but they're short, and they
Yeah.
just kind of like, think about it like it's a tube, right? It's got muscles at the anterior end where it starts.
Yeah.
Then the muscles contract, and it pumps the blood. And it's kind of a muscular part, and then it pumps the blood through vessels. But then the vessels end, and it's because it's not even blood. It's called hemolymph. It's the blood and interstitial fluid. It's just one thing because it's an open system. So the blood just kind of comes out of the vessels and it kind of falls down. And then when the heart relaxes, that kind of sucks the hemolymph back up through the tissue back into the heart. And it's an open system. Structurally, it's completely different from a human heart. I would think that it ages because it's, an animal, so it's going to age over time, and that is expected because that's what all animals do. But structurally, not very similar. I mean, it's contractile tissue, so that's going to be somewhat similar.
Yeah,
Oh,
I mean, it's muscle. There you go, guys.
right. I will say human cardiac muscle is super, not human, but like animal cardiac muscle, super, super specific because it's autorhythmic, so it triggers its own contractions, and it has connections between the cells so that you can have the action potential move smoothly from cell to cell. I don't know what the physiology of insect hearts are.
I'm not sure either.
But yeah.
I will say though there does seem to be the possibility that there is some small degree of homology there. You can actually kind of think of deuterostomes as being backwards, upside down
Yeah.
stones, which is one of the
Yeah.
reasons why like the central nerve cord for insects is on the ventral side as opposed to the dorsal side, and their heart is on the dorsal side as opposed to the ventral side. So there is a chance that there is some kind of homology way back at like, Urbilitarian. Maybe Urbilitarian had a little nerve cord and a little muscle that pumped something, but that's still a pretty big supposition. Like maybe.
Yeah. I could not say. Yeah, I don't know. I don't know enough about the ancestral coelomate, right? The most recent common ancestor coelomate. I don't know enough about what we think
No
that
one
thing
does.
was to. Yeah, right.
So
So,
there's two big competing hypotheses, which is simple versus complex, and there's not great supporting evidence either way. The simple would mean that a lot of these features are derived analogously, and the complex would mean that a lot of these are probably homologous. So, but which one of those is still a very open question. There's really no consensus. But anyway,
just one more thing on that. In either case, I would think that you would have deep homology at the genetic level for a lot of them. So when you look at things like, right, so like for legs, eyes, like we know the genetic basis for eye development, for leg development across all these different groups. And we know that common ancestor didn't have legs, but it's the same genetic networks that are operating across all these different ones. So I wouldn't be surprised if it was something like that.
So then we have where the biggest similarity is that both have heart disease as a result of getting older. I mean, if that's your biggest similarity, I don't know that I would call that very little difference, but sure, whatever, guy. Studied flies in order to produce cardiac medications. That's a whole sentence. I feel like it should have a subject.
There's a translation issue here, but that we get the idea.
Okay. Professor Juan Alvarez Bravo at the University of Tokyo, which is not where I would expect someone with that name to be studying, but that's okay, mentioned that this is the last one can accept the flies in a hospital. But soon we will witness a rapid treatment for many diseases which consists of extract from flies. There is also some researchers in the US dedicated to find new treatments using flies, and they confirm that treatment with the use of flies is scientifically accepted in the near future. Some researchers at Auburn University tried to find a protein in the fly's saliva. This protein can accelerate the lengthy process of healing wounds and chronic skin cracking, and not too long ago declared the researchers at Stanford University that the first found of a substance in the fly that can improve the immune system of a human being. Moreover, the researchers also found that the best way to release these vital substances is to dip the fly in water because these substances are concentrated on the outer surface of the fly's body and wing. The aim of this research was to clarify the miracle in the Hadith of the Prophet Muhammad, peace be upon him, by studying the antimicrobial effects of the fly wings, and to understand this aspect from the scientific point of view. So actually, I think it might have been easier for me to read this in Arabic, because it heavily reads like someone who has a little bit of English, but who forgets that the grammar is different, and you're going to need to do things like not use the verb first in a sentence and explicitly give a subject.
So the thing I want to point out here with all this background, which the degree to which it's relevant, fine, whatever. Some of it doesn't matter, like the fly lifespan, but like this stuff about like reference 3, for example, the flies being used as treatments for different things potentially. These are not like scientific papers. That's from an article called "The New Buzz on Antibiotics" from ABC, which is to say the news network, right? This is not like a scientific paper that we're referencing to bolster this. This is just kind of general news items. It looks to be the same case for reference 2. Looks like 5 is an academic source, but there's no journal or anything mentioned. Science Daily is kind of a science publication, but it's not peer reviewed, right? There's a bunch of like, these are not peer reviewed papers. And typically in the introduction where you're going to set up a work like this, you need to lay out the state of the field using the existing research, like literature, to set that foundation. And that's not done here. That's irrelevant to the validity of what the claims are going to be, but just in terms of like setting up the argument, that step has not been done.
Yeah. It seems like there's just been sort of a hand wave towards like, people are researching flies for medical use. And it's like, okay, I mean, people are researching almost everything for medical use, so that's not surprising. Because, I mean, one of the things is, who knows where the next big treatment for something is going to come from? Because it's pretty unpredictable. So you study as many things as you can. So do you want to get into the materials and methods, or should I continue?
Let's do it. So this is where I started just highlighting a lot of things because, as the saying goes, I got a lot of problems. So we start with collection of flies. 4 flies, Musca domestica, were collected. The left and right wings for each were separated by sterile forceps. Okay, first problem, we can just stop right there if we want, because if your N is 4,
you're not generating any statistical significance.
That's the end, right? We're not, that's the first thing. Anybody reviewing this paper would get to that line, which was actually referenced up in like the abstract, I think. So you get that far. As soon as you get to N equals 4, like whatever you're about to tell me, it doesn't matter. Go repeat it, get, it's flies. So like this is easy. Get 20, get 100, right? Like it's easy to work with flies. They're fast. So
if you can't get more than 100 house flies, you're just not trying.
Yeah, so that's the first problem. You gotta have a larger sample size than 4 in order to have any confidence in what's going on here.
Yeah, this is supposed to be a regular study, not a case study. So
yeah, this is like a very straightforward, like experimental. We're going to do this. We're going to do that. We're going to compare the results. We have a hypothesis about what the results could be. It's very straightforward. But if you're doing comparisons like that, you can't have it. You can't do it with 4. You need a lot. You need a lot. That's the first thing. We need more data. So preparation of wing extract, each fly wing left and right dipped in a sterile labeled test tube containing 5 ml of sterile nutrient broth media purchased from Whiten BioLife Company, produced by, is it Jaleel Medical's company? Okay. Sure, I don't have any problem with any of that. That's, you're using whatever media, that's fine. This section, microbiological examination, this is where I just highlighted the whole 3 paragraphs. The preparation of both wings extract was incubated for 48 hours at 37 degrees Celsius. First problem, we're looking for bacteria by incubating them at 37 degrees Celsius. These are flies. Flies are ectotherms. 37 degrees Celsius is great if you're looking for human bacteria, because that's our body temperature. So anything that lives on or in us is adapted to 37 degrees Celsius. But that's not the case for flies. So the conditions to grow any bacteria that would live on the fly are wrong, because they're ectotherms. They're going to be whatever the ambient temperature is.
Now, I do have a question, because I live someplace where 37 is actually a pretty normal ambient temperature.
Oh, that's a good point.
So presumably those same flies would have, bacteria that could grow reasonably well at that. So I do think it is a question. Maybe we should use different temperatures. But I would say that 37 doesn't seem like a temperature you shouldn't use. I would like to see different temperatures used in an experiment with more groups.
I think that is a better critique than mine. You're absolutely right. I totally didn't. And here I am sitting here. It's actually like 30 something right now as I sit here. So it's actually like mid to high 30s is rare for me, but it's not unheard of. For you, I'm sure it's more common.
Look, it gets into the mid to high 40s here.
Yeah, so that's not acceptable.
We're currently at the low 30s right now. but
Yeah, that's about where I am.
by the end of the day, I'm sure that we will get around 36-ish.
Okay, so you know what? That's a fair point. I withdraw my objection. My new objection is what you said, which is that we should have tested a range of temperatures.
Well, we only have 4 flies to work with.
25, 30, 35, 40, right? And, you know, right. So I think you get a bigger sample size, you test a range of temperatures. There you go. Okay. One loopful from each test tube was examined microscopically. So I want to explain what this is. So a loop refers to an inoculating loop, which is a little twist of, it's either going to be like plastic or metal, and you put it in a solution where there's bacteria. And you pull it out, and it's like you know, blowing bubbles, right? It's like a little loop with some liquid in there, right? And like you would blow bubbles through, but it's tiny, and you could actually calibrate them for different volumes. So you could get like a 1 microliter inoculating loop, so you get just one little tiny bit of fluid in that little loop. I suppose you could examine those microscopically, like you could look at the fluid in the loop under a microscope. I've never done that. I've never heard of that. This is the first time I've heard of that being done. My experience with microbiological techniques is more virology oriented. So it's possible that this is a common thing that I'm not familiar with. But I did do a fair amount of work with like bacterial cultures because I had to grow my bacteriophages in bacteria, and doing things, you know, like counting colonies and classifying whatever your bacteria are and everything. And I've never seen that done. It's not to say it isn't done, not to say it can't be done, but that is an unusual way for me, in my experience, in terms of like, are there bacteria present in the liquid, to look at the inoculating loop under a microscope.
Yeah.
Right. So that strikes me as odd. So for investigation,
odd, but not necessarily like an invalidation.
Yeah. I mean, they did other things. So the other things are the important part, and we'll get to those, but it's just struck me as odd that was one of the tests. So so they examined microscopically for investigation of microbial presence, bacteria, as well as fungi. For further microbiological examination, 1 loopful from each test tube was inoculated in 2 sterile labeled Petri dishes containing nutrient agar media and potato dextrose media, purchased from Whitetown BioLife Company, produced by Jalil Medical Company. So okay, so what we're doing here is we're taking the fly wings, we're putting them in liquid media, right? Mixing them up. And then we're going to take some of that media with a little loop, and we're going to put it on a Petri dish. If you've ever gotten like a throat culture for like check for strep throat, where they stick that big Q-tip down your throat and then they wipe it on the plate real fast, that's what we're talking about here, except you're using a little loop to do it instead of a Q-tip. Same idea. You're taking the plate with the medium where the bacteria can grow, and you take a little bit of this liquid and you spread it on the plate, and the goal is to see, do bacterial colonies grow? So then the cultivated plates were incubated for 48 hours at 37 degrees Celsius for the bacteria and 25 degrees Celsius for the fungi, and then the plates were examined for bacterial as well as fungal growth. And now we get into the results. It's one specific paragraph here, and it references this one figure that is the rest of this page. So they write, the cultivated plates on nutrient agar media and potato dextrose agar media demonstrate both bacterial and fungal growth for the left wing's extract plates. However, there were no bacterial or fungal growth for the right ones, as shown in pictures 4 and 5. So we could go and look at pictures 4 and 5.
Which are on screen now.
And the first thing I want to point out here is that these pictures are not going to fly as like reliable results, because first we have an n equals 4, but we're only seeing one of each. We should see at least 4 plates, right? We should see all the right, all the left plates. We want to be able to do a 1-to-1 comparison for each pair. So that's first. Second, the picture quality is just like not good enough to see what's going on here. But what it looks like they're showing is that on the left plate, you have this big, it's circled and it has the arrow pointing to it, this big white mass that they're saying, okay, that's bacterial and fungal growth. And then on the right plate, it's clear, you don't see any bacterial or fungal growth. So that's taken as evidence of the hypothesis that there's stuff growing on one wing, but not the other wing.
Actually, it occurs to me, we shouldn't just see 4 left and right sets. We should see 8, because they incubated separately for bacteria and fungi.
Oh, you're right. Yeah, we should see 4 pairs for bacteria and 4 pairs for fungi. You're absolutely right. Yep. So here's the problem I have with this left plate. So I'm actually going to share my screen real fast because I'm going to show what it's called a streak plate when you inoculate a plate with a loop. Do some streaks, and then typically what you do, the goal is to get individual colonies. So you do some streaks, then you turn it a little bit and like pull out of there, and then you turn it again and pull it. So you're trying to get individual bacterial cells to spread out to get colonies. And so I'm going to share what this looks like, which is this right here. So this is what a streak plate looks like. You start over here with the loop and you go back and forth. And then, can you see my cursor?
I can.
Okay, great. So you start over here with the loop, you go back and forth, and then you pull a little bit out and you go this way, and then you go that way. And then by the time you get over here, you see you get these individual colonies, right? So you could document like, okay, there's bacteria here, and you could pluck an individual colony and say, okay, let's characterize this thing. What in the world are we dealing with? So that's what a positive result for bacteria would look like here. Now, that's not what we see in this paper. We see this big mush of white stuff. And as someone who has contaminated a whole bunch of plates in my career working on bacteria microbiology, you screw up plenty often, and you do it enough times that even if you only mess up 1% of the time, you still contaminate a bunch of things here and there. And so let me show you what a contaminated plate looks like. This is a plate that has a number of different contaminants on it. So let me just share this real fast. And while I'm sharing this, I do want to note that this is from Wikimedia Commons. It's from a user called Garnhami, and it's shared with a Creative Commons 4.0 license. So here is various fungal contaminants on this plate. It also looks like there's some bacteria, like right there, but like these big white things, that's fungal contamination. This stuff over here, that's all fungal. And that's what the plate looks like. It doesn't look like a loop inoculated streak plate. It looks like a big mass of fungal contamination. So I'm like 80% sure that whatever's growing on that plate didn't come from the loop. It came from contamination over the course of this experiment.
Right. And with an N of 4, that's, if you're trying to do statistics, one instance of a contamination is just going to completely throw it. Like that's
right.
What is that? It's going to be an eighth of your results is just one contaminated plate. That's
ridiculous. And also, we don't know whether this was supposed to be a bacterial or a fungal plate, because it says bacterial and fungal growth. I mean, that sure looks fungal rather than bacterial, but we don't know if that's the plate that was grown at 37 or at 25, right? We don't know what that's supposed to be. But again, as someone who's done this a lot, as someone who has checked their plates in the incubator at 9 a.m. and gone, shucks, I messed up yesterday because I had this big white mass of contamination there. You know, I've been there. That's what that looks like. That looks like fungal contamination.
Well, this is something involves something I want to point out about how scientific papers are supposed to work, right? You need to show all of your work. The raw data should be available in your paper. It doesn't necessarily have to be in the body. It can be in like an appendix or something, right? But the raw data should be there so that anyone else can crunch your numbers independently of you and hopefully get the same results that you get. Because if they don't, then that means that either that one of you has made some kind of just basic math error. And so this paper doesn't meet that. And I think for that reason on its own, it shouldn't have been published, at least in this format. Like it should, should have been sent back and said, hey, you got to show all your work.
You need the supplementals to be like, okay, we're showing a representative. It's perfectly fine to say, I'm going to show you a representative example of a positive fungal plate and a negative fungal plate. See, supplemental 2 for every plate we did, right? And you have a file, with just all the images of each pair of plates.
Which we don't have in this because we've both read all of this.
Which we don't have.
So yeah,
we do not have that.
Yeah. So I just want to say, we have both read through this paper before. This is, we're not coming at this cold. So we do know that this supplemental data just isn't there.
I don't have it. Yeah. Okay. So that's the plates. That's pictures 4 and 5 in figure 1 right here. But let's talk about pictures 1, 2, and 3, because this is also a problem. So these are pictures of presumably one pair of the test tubes with the liquid media that they mentioned back up in the methods, right? And what we see here is for the left one, in picture 1, it's labeled turbidity, and there's something in there. I can't really tell what it is because, again, the picture quality is just not sufficient to be able to see what's going on here. And then on picture 3, we have the right one, which presumably shows none of that. And then side by side, you see the two of them. Now, the problem here is, well, there's a couple problems here. One is this is like, you need to, if you're going to show these pictures, you need to have a specific background. You need to like have all the pairs. It's the same problems with the plates with picture quality, picture quantity, and then show all of them either directly or in a supplemental, right? We can't draw any conclusions based on one of these without seeing all of them, because we only have a sample size of 4, which
Now we're
we
reducing
said,
it to
okay,
1.
Right. So we get 8 comparisons, but we're only showing one of those comparisons. The second problem, and the larger problem here, is that this is not how you measure turbidity. You don't hold it up and eyeball it. What you do is you take a little bit of this and you put it in a tiny little tube and you put it in a device called a spectrophotometer that can actually measure the opacity by measuring the absorbance of that solution. So it'll shine light at a specific wavelength through that solution, and it will measure how much of that light is transmitted through it versus how much of that light is absorbed by the solution. And this is how you quantitatively evaluate the turbidity of a bacterial culture. You measure the absorbance, right, using a spectrophotometer. So you should be able to take a bit of those, or if you're doing fungal as well, 8, right tubes, and show that they're not different from a negative control. And then the left tubes, you should show some statistical significance in terms of the level of turbidity compared to the right tubes. That's not what's done here. Just eyeballing it.
Yeah. The other thing is also, I would like for proper experiment construction to also see tubes and plates from the sterile medium that were not did not have a flywing dipped in, because that would be the proper control. There's
always have to have that control.
Yes. Yep. So now I can imagine that the controls would probably show essentially no fungal growth or bacterial growth. However, if they did, then we could say, well, clearly your controls in your lab were insufficient to prevent contamination, so that's the reason we're getting results.
Yeah. Exactly.
We don't have that, and they don't even say that they did it. So this experiment doesn't have proper controls.
Yeah.
I think that's a big problem, because you need the controls.
That's a huge problem, because how can I evaluate left versus right when the claim is that the right shows nothing and the left shows something, when we don't know what absolutely nothing looks like?
Right.
We don't know what this media looks like without any treatment at all. And that's a problem. So going back to measuring these tubes with a spectrophotometer, you got your negative control. Presumably you've got a positive control, right? I'm going to take a colony of E. coli. I'm going to put it in the media. I'm going to grow it up overnight, right? And we're going to measure that. So you've got a positive, you've got a negative, and now we've got our left and right, and now we can test those and see how they shake out.
Yeah, this comes across more like a high school science fair experiment than an actual experiment done by a real scientist in a proper microbiology lab. And now I'm not saying there's no value to doing that kind of thing in high school, but there's a reason that high school science fair projects don't tend to get published.
Yeah. And I mean, this is the kind of thing you could do. Like, this is like, when I was in grad school, you'd be like, I wonder. And then you just take 3 hours and just do a quick thing once, right? And be like, did it kind of work? Is it does the technique work? And you're not going to get real results. You're not going to get reliable results. But you could just see, did stuff grow? Did stuff not grow? Is this worth spending a week, doing for real correctly? Right.
It's sort of like the pre-experiment before you do the real.
Yeah. Just does the technique work in principle, right? And you do it you have a very small N, maybe the controls are there or not. It's just you just see if it works at all, just at a basic level, and then do it, for real after that.
Yeah.
But get the techniques down first. And here we're still at the get the techniques down state of it. You can't get reliable data at that stage with no controls, with small N, with again, I just want to emphasize, this is not how you measure, is there bacteria growing in a test tube, right? You've got media in a test tube. There's a way to measure, there's a way to answer the question, is bacteria growing in that tube? This is not how you do it. You don't look at it up against the background. You don't put it next to another tube. You don't look at it under a microscope. You put it in a spec and you measure it against a positive and a negative control. And then you can tell if there's bacteria growing in it.
That's a lot of work, though.
It is. And to be fair, it requires some specialized equipment, right? But you have to be precise about these things. You can't make claims that have medical significance without dotting your I's and crossing your T's.
Yeah. Well, now I think we're at the discussion.
So the discussion I'm happy to go through it. I don't have a whole lot to add because it's all about for me, it's all about the methods and the results of like, this was not done correctly. And whatever the for people who are not familiar with scientific writing, the discussion is where you frame the importance and the implications of your work in the broader context of the field. And in order to do that, you need to have results that are reliable, right? If the problem is these results are not reliable, then any broader conclusions, any broader implications you're going to draw from that are necessarily going to be questionable at best, right? So that's kind of where we are now. We've gotten through this. The methods are insufficient to address the question, right? We had, to the author's credit, we had a specific hypothesis, right? Great, this is extremely testable. The methods are not sufficient to address it, and the results as presented do not provide a reliable answer to the question that was asked. So now we get to the discussion. We want to talk about the implications of this going forward. What can we say that's reliable?
Yeah. Well, there's a little bit I do want to talk about in there. Not much. I don't think we're going to go through the whole discussion because, like you said, there's most of it's irrelevant since you haven't actually demonstrated any real effects of fly wings. So I'll read a little bit of it. In the latest scientific research, scientists have proved that flies, the greatest prophet told us about the presence of cure in its wings. This has certainly proved the truth of this prophetic speech. Okay, in addition to being a hard-to-parse sentence, I think what we're saying is that, you know, this being some of the newest scientific research on fly wings has more or less confirmed what Muhammad said. Now, I will say, if the results as presented were actually done in a way that had some statistical significance and had proper controls and were done with actual proper methodology, then yes, that would be something that would lend credence to the original claim from Muhammad. Now, okay, look, I do know that there are some questions as to how authentic all of these things are, but I'm just, like I said before, we're just accepting it's Muhammad.
Right.
Who cares? But we're skipping down a little bit. This hadith means that the fly carries on one of its wings a disease and on the other a cure for the same disease. Now, one thing I want to point out about this is that this experiment, even if the methods were good, wouldn't actually prove that you could use this wing to cure a person from this disease. So that's an important thing, because the point of this hadith is supposed to be that whatever the cure is going to dissolve into your drink because both wings have now been dunked in. And so both the disease is going to get it spread out through your drink, but so is the cure, so it's going to be okay. Whereas if you don't dunk the fly in, who knows, you might have only gotten the disease wing, right? That's the basis of this. But even if it were true that you couldn't grow these fungal or bacterial pathogens from one wing versus the other, that doesn't mean that you could then cure this disease, because how do we know that this, whatever is preventing the growth on one of these wings, is something that's actually going to dissolve into your drink in sufficient doses? No, that would have to be something that you would have to test separately. So we're only testing the first part of this. Is there a difference in fly wings? We haven't even actually gotten a yes answer. But then if we did get a yes answer, we would then still have to go further and say, okay, is there actually some kind of medical value to having both wings be dunked in a drink? So first you'd have to say, do fly wings falling into drinks cause disease in any measurable amount? That's an open question. Then once you get a yes to that, then you can go ahead and say, okay, does putting both fly wings in a drink prevent that increase of disease incidence that we saw in the first or the second experiment, I guess, at this point? Mm-hmm. Then finally, once you get a yes answer to all three of these, then you can say that this claim has been confirmed. But we're not even at step one.
And even once you get to there, there's the open question of broader medical applicability. Because back in the intro, the author said this is, well, we can maybe use this to cure like these various pathogenic bacterial infections.
Yeah. That, I would say that is a step beyond just the claim from the hadith, but it is an interesting question.
I think that's fair, yeah.
I would say if you can just answer yes to all three of those, then at least you've confirmed the hadith, which does seem to be the biggest motive. I mean, it has the word miracle in the keywords.
Right.
So clearly this is a paper that is saying, okay, Islam is true. Here's a claim from Islam. If we can prove it, that will help convince people that Islam is true, or help keep Muslims convinced that it's true if they're already convinced. And I'm not here to try to disprove Islam. I don't care to disprove Islam. It's not my point. But what I am going to say is this particular claim is scientifically testable. This is an insufficient test of it. And even if it were a sufficient test that had the results that the author wants us to say, or wants us to believe that it had, still wouldn't actually be sufficient to confirm this experiment. I think it's time for some final thoughts, because I don't think there's much else in the discussion that I really care about.
Yeah, I think that summarized it nicely. I think the big takeaway here is we have a very straightforward, testable hypothesis, right? Some kind of bacteria or fungus on one wing, not on the other wing, and specifically the something to kill or inactivate those microbes on the other wing, right? We have this is very testable. This is pretty straightforward from a microbiology perspective. You can test that, right? The experiments done here and the data shown here are not sufficient to draw any conclusions about the validity of that hypothesis, right? They just are not up to addressing that question. We need larger sample sizes. We need more precise techniques for determining the presence or absence of microbes, whether they're fungal or bacterial. We need just to quantify the results, how much, if any, right?
There's a total lack of math in this.
Yeah, and I would speaking in my experience, what I would want to see is okay, you had this level of turbidity in this, you had this level of turbidity in that. From each of these cultures, we took a loop, we put it on a plate, and we documented, X number of what's called plaque forming units. Or sorry, that's a virus term. Sorry, I slipped into virus, Dan, for a minute there.
How dare you?
Colony forming units, because we're dealing with bacteria, right? So you would document basically by diluting it many times. So you do like a 10-time dilution iteratively, right? It's called a serial dilution. And then you put it on the plate, and at whatever level of dilution you're able to count the number of individual colonies, you can then just do the math to work backwards and calculate your population density for your liquid cultures, right? So you can actually quantify how much bacteria is in there, not just say it's present or it's absent, but like how much is there? How fast did it grow? Did you inhibit it?
Math in a scientific experiment.
Right, but look, one of the major claims is not just that it's not present on the one side, but it's inhibited. So you could do these growth experiments where you'd say, okay, here's one wing, right? There's bacteria, here's the population density. Here's the other wing, there's nothing. Here they are together. Let's see what the rate of inhibition is, right? To actually verify that claim. That's how you would do that. No attempt to do that here, right? So my big takeaway from this is that there are specific microbiological techniques that we could use to test this hypothesis, and that was not done. So we can't say anything about the validity of this hypothesis because it's just simply in this publication, there's no indication that it was actually tested.
I think my big final thoughts on this are, look, I think that with some modification, this paper could become the basis for a future paper that's worth something, right? Do the real controls, actually show all your work, do the real measurements, and if it still supports this hypothesis, then that's great. That's an interesting thing. I would love to know about that. It'd be really weird, very unexpected, because quite frankly, the claim is a bit outlandish, to put it mildly. If I'm being more serious, I would say it's laughably ridiculous. That's my actual opinion on this hypothesis. However, like Dan said, it's very testable. Just please test it. You have to actually do the test. The only other thing I have is this is one of only 2 papers that I was sent. And I'm going to try and set up another discussion on the second one. And the second one is on the salubrious effects of camel urine for human beings. Because famously, Muhammad also recommended to various people that suffering what was basically probably some kind of heat exhaustion or possibly even early stage heat stroke, that they go into the shade and drink camel urine. So we're going to take a look at how healthy it is to drink camel urine. And I do want to point out that not too long ago, the World Health Organization put out a statement, which was they tried to publish in the Middle East, urging people to stop drinking camel urine. So right now, the WHO is anti-camel urine drinking, for whatever that's worth. Put that out there. But that is for perhaps another, I don't know what we're going to call this, Islam paper reading time? That's a terrible title. I'll come up with a better title. All right, thank you, everybody. And Dan, is there anything that you always have stuff coming up on your channel? There's always gonna be something.
So I have some stuff coming up. I'm working like literally right now today. I've been working on a collaboration with our mutual friend Jackson Wheat, so I'm excited for that. He's been very patient, waiting for me to get around to it in between things like summer classes and children. So I appreciate his patience, but eventually in the near future I'm going to have that done. So look out for that on Jackson Wheat's channel soon. I've got a couple other collaborations on the horizon that are coming up. And as always, I'm going to try to do stuff. I just put out a video as we sit here recording this. I put out a video yesterday on the use of Darwinism by creationists and why that's not okay. And I try to keep up on the latest on what creationists are doing and talk about why it's wrong. And I'm sure if you hop over and subscribe, you'll see me doing stuff at a sporadic at best schedule.
All right. Well, thank you very much. And I just want to say, please, if you like this video, hit the like button. Make sure you subscribe. Hit the bell icon. Make sure that you're always notified when there's more Dapper Dino content. Every like and subscription really does help. And with that, I'm just going to say thank you to my patrons and channel members, and we're out of here. I just want to take a second to thank my channel members and patrons, especially those pledging $20 and above. Bob Knob, Benthoven, Ian Chen, Sphincter of Doom, The Venerable Bede, Patrick Dennis, and Res Instance. My channel members and patrons help make this channel possible, and without their support, this channel really wouldn't exist. If you would like to help support the channel as a member or patron, there are links in the description to both join the channel as well as join the Patreon. Patrons and members get mentioned in these credit scenes, as well as getting early access to my scripted videos, and if you pledge $10 or above, you can also get access to various 3D assets that I create for Blender, both for use in the channel, as well as just general giveaways to my supporters to help in any Blender projects they might have. If a monthly or annual pledge isn't for you, then there is also a merch store linked in the description, and if none of that's right for you, please just like and subscribe because every like and subscription really helps the video out. Thank you.